So this video is going to talk about how Nirenberg and Matthaei and Nirenberg and Leader's experiments have helped us crack the genetic code. So Nirenberg and Matthaei took a poly-U, which was just uracil nucleotides, so the only codon available in the experiment was UUU, a cell-free extract, and 20 amino acids, one of which was radioactive, and put them into a tube. And they allowed the tube to be incubated for translation to occur, and then they filtered out the protein. And if the protein was radioactive, that meant that whatever amino acid was radioactive in that tube coded for that codon. So they repeated the experiment in 20 tubes with a different amino acid being radioactive each time, and they found that when UUU was produced radioactively, it was produced in the tube that had phenylalanine radioactive. So then they repeated the experiment with a poly A, and that was connected with lysine, and a poly C that led to proline. So they found 3 of the 64 codons, and then Nirenberg and Leader wanted to expand upon the knowledge of how codons and amino acids were connected. So they took short mRNA sequences whose sequences were already known and put them in a mixture of tRNAs connected to amino acids, and that's shown here, and that's going to be this in our model. And they also had ribosomes in the mixture. So the mRNA codons bound to the ribosomes, and that's shown here. The green is the ribosomes and the blue are the mRNA, and that's going to be this structure. So the mRNA specified for a certain tRNA for it to bind to because it's an anticodon and codon. So they complementary base paired and formed this mRNA bound tRNA molecule. So then they put the mixture through a filter, and there were tRNAs that weren't bound to an mRNA ribosome molecule, and those passed easily through the filter, the unbound tRNA. But if the tRNA was bound, then it couldn't pass through the filter. So they could look at what molecules were stuck in the filter and figure out that for this codon that it coded for valine, and they were able to use this process to figure out the other 61 codons, and that helped us figure out the entire genetic code.